Hello everyone, right now we have photosynthesis in higher plants chapter. Initially we have the reaction here. 6CO2 plus 12H2O in the presence of chlorophyll and light gives C6H12O6 plus 6H2O plus 6O2. Photosynthesis is a redox reaction and oxidation of water takes place and reduction of carbon dioxide takes place. Oxidation of water and reduction of CO2 takes place. Photosynthesis is anabolic and endergonic process. Anabolic and endergonic process. And next, Joseph Priestley. Animals die due to suffocation as burning of candle and breathing of animal damages the air. Animal die due to suffocation as burning of candle and breathing of animal damage the air. Next, plants restore to air whatever the breathing animals and burning candle removed. Okay, so next, Jan Ingenhouse in 1779, he concluded that light is essential to the plant process that somehow purifies the fouled air in Priestley's experiment. Next, he also observed that in the presence of sunlight, aquatic plants release some gas. Later, the gas was found to be oxygen. Next, he showed that only green plants release oxygen. Van Niel studied the photosynthesis in green and purple bacteria and conclusions. Hydrogen comes from the suitable oxidizable compound and reduces CO2. Next, in bacteria, H2S donates the hydrogen for CO2 reduction. In green plants, H2O donates hydrogen for reduction of CO2. Oxygen evolved by plant comes from water. And oxygenic photosynthesis occurs in bacteria. Next is given the structure of chloroplast. In it, the outer membrane, inner membrane, stromal lamellae, granum, and then stroma, and then stroma is a site for for dark reaction, and then the ribosome, which is 70S, and then starch granules is given, and one single circle denotes thylakoids, which is a site for light reaction, and many circles denotes a stack of circles that denotes the granum. Okay, so next thing we have early experiments. Mole's half-leaf experiment. It showed that KOH in the tube absorbs all the CO2. The portion of the leaves in the test tube shows negative test for starch. Conclusion: CO2 is required for starch synthesis, that is photosynthesis. So Mole's half-leaf experiment concludes that CO2 is required for starch synthesis, which is photosynthesis. And next, variegated leaf experiment. Part of the plant covered with black strip to avoid light exposure, and covered portion of the leaf shows negative test for starch. Conclusion that light is essential for starch synthesis, which is photosynthesis. Next, Priestley's experiment. When when the mouse is kept with the burning when the mouse is kept, that time candle burns, and the mice is when the mice is dead, the mice dies due to the suffocation as the burning candle and breathing animal release CO2. And then when the mint plant is kept, then three of them can survive. Next, T.W. Engelmann's experiment. Growth of the aerobic bacteria was high in blue and red region. This indicates the release of more oxygen in red blue light. Next we have cladophora, green algae. Next is the absorption spectrum and action spectrum. Absorption of light by chlorophyll A is maximum at the blue and then in red wavelength, and in action spectrum the blue red light, uh, the rate of photosynthesis is maximum in blue and red light. Rate of photosynthesis and oxygen evolved on the y coordinate. Here you have to remember the carotenoid the curve of carotenoid, chlorophyll A, chlorophyll B also. Next is dependency of dark reaction on light. Photosystems. Chlorophyll A is the chief pigment for photosynthesis. PS1 is 700, P700, PS2 is P680, and PS1 present in the non-appressed part of the thylakoid and the stroma lamellae. Stroma lamellae lacks NADP reductase enzyme and PS2. Chlorophyll A is called reaction center. Chlorophyll A is called reaction center, and accessory pigments forms the light harvesting complex LHC. Stroma lamellae lacks NADP reductase enzyme and PS2. Chlorophyll A is the reaction center. Light reaction. Light strikes the leaf and then mesophyll cells and then chloroplast to thylakoid membrane, then to photosystems reaction center. Then electrons from the PS2 P680 gets excited. First electron accepted from PS2 is pheophytin, then electron moves to the ATS and reach PS1. Meanwhile, H plus from stroma are influxed to the lumen of the thylakoid. Photolysis of water takes place. Hydrogen increases in the lumen. Water provides electron to PS2. Electron from PS1 gets excited and reduce the NADP plus to NADPH. Concentration of H plus increases in the lumen. Proton gradient is generated across the thylakoid membrane, and H plus from the lumen diffuses to the stroma through ATP synthase. ADP to ATP is formed through photophosphorylation. Then here Z scheme is given, PS2, PS1, and next cyclic photophosphorylation. Only PS1 is used, only ATP is synthesized, and no NADPH is formed, no oxygen is evolved, and occurs in stroma lamellae. Next, this is non-cyclic photophosphorylation and chemiosmosis. For one CO2, next is C3 cycle, dark reaction or biosynthetic phase, RUBP to RUBP to 3 phosphoglycerate through carboxylation, then 3 phosphoglycerate to triose phosphate through reduction, triose phosphate to RUBP through regeneration. In carboxylation, RUBP Rubisco is used, and in reduction 2 ATP and 2 NADPH are used, and in regeneration 1 ATP is used. So for 1 CO2, 3 ATP and 2 NADPH are used. For 1 glucose, 6 CO2, 12 NADPH and 18 ATPs are used. Rubisco shows carboxylation activity for the CO2 succession, and ATP and NADPH are supplied from light reaction Oxidation of 1H2 is required for the formation of 1 NADPH, hence 12H2 are required for 12 NADPH. It was first discovered in chlorella, a green algae. Next we have three steps of C3 cycle. First is carboxylation, which is the most crucial step, and enzyme involved is rubisco. First stable product is 3-PGA, which is 3 carbon, and rubisco binds to CO2. Next is reduction. It is a multi-step reaction, and 2 ATP and 2 NADPH are used for fixation of one CO2 molecule. And next is regeneration of CO2 acceptor RUBP, which is 5 carbon. 1 ATP is required to regenerate 1 RUBP. 1 ATP is required to generate 1 RUBP, and carboxylation is the most crucial step given. Next, C4 pathway or Hatch and Slack pathway. C4 plants, they can tolerate high temperature, show response to high light intensities, and next, do not show photo respiration, and they are more efficient. They have low CO2 saturation point. In C4 plants, for synthesis of one glucose molecule, total 30 ATP and 12 NADPH are required. 30 ATP and 12 NADPH are required for synthesis of one glucose molecule. And next, phosphoenol pyruvate is the first CO2 acceptor. First CO2 acceptor of C4 plant is PEP, and first product form is OAA, which is 4 carbon, and PEP is 3 carbon. Next, C4 plants examples are maize, sorghum, sugarcane. Optimum temperature is 30 to 40 degrees Celsius. And photorespiration, which is C2 cycle or glycolate cycle. So rubisco binds to oxygen with RUBP. 3 carbon, which is PGA, and 2 carbon phosphoglycolate are formed. Neither synthesis of sugar or nor ATP and NADPH. Loss of carbons in the form of CO2 at high light intensities and high temperature. In low CO2 and high oxygen concentration, C3 plant shows photorespiration, low CO2 and high oxygen concentration. Okay. Next, organelle involved are chloroplasts, peroxisomes, and mitochondria. Then we have mesophyll cell, granule thylakoid, and bundle sheath cell. Large number of chloroplasts. Walls are impermeable to the gaseous exchange. No intercellular space. A granule chloroplast site for C3 cycle. Factors affecting the rate of photosynthesis. Blackman's law of limiting factors. Rate of chemical process is determined by the factor which are near to the minimal values. And internal factors are like number, size, age, orientation of the leaf, mesophyll cells, and chloroplast. Internal CO2 concentration, amount of chlorophyll. These factors depends on the growth of the plant and genetic predisposition. Next we have external factors, which are first is the light quality, maximum photosynthesis at blue, red, and PAR is 400 to 700 nanometer. Second is the quantity. At high light intensity, rate does not show further increase as the other factors become limiting. And at low intensity, rate of photosynthesis is directly proportional to intensity of the light. The graph is given here. And next, CO2, rate of photosynthesis directly proportional to CO2 concentration, but only up to 500 ppm, beyond which light becomes damaging. Current atmospheric CO2 level is limiting for C3 plant but not for the C4 plant. Saturation of C3 is 450, beyond 450, and C4 is 360. CO2 fertilizing effect, high CO2 concentration, higher yield, example tomato. Temperature, dark reaction is more affected than light reaction because of the bioenzymatic content. And next, optimum temperature for C3 is lesser and C4 is more. Water, indirect impact on rate of photosynthesis. Water stress, the stomata are closed and low CO2 availability, low rate of photosynthesis. And in water stress, wilting of leaves, reduced surface of the leaves, low metabolism, low rate of photosynthesis. Last is the summary. During the photosynthesis, CO2 is used for making carbohydrates, principally glucose and starch. Within the chloroplast, the dark reaction occurs in stroma. And a correction in summary: NAD is formed from NADH, but it is NADP is formed from NADPH. Rubisco always catalyzes a wasteful oxygenation in reaction in C3 plant, which is photorespiration. Thanks for watching.